We are very happy to have with us today Mark Shein-Idelson. He has a background in physics and electrical engineering, and he's doing his PhD with Ben Yaakov and ELSC-ICNC at Tel Aviv University. He has the final stage of the PhD, and he's doing exciting work about in vitro after experimental work on inhibition, excitation, balance, networks. You'll tell us something about the computational aspect of this. Thank you.
Okay. Good afternoon, everyone. Can you hear me? Yeah? Okay. So thank you for inviting me. And what I want to show you today is an experimental work that I did in a joint lab of Professor Eshel Ben Jacob and Yael Hanin from Tel Aviv University. And our aim in this work was to examine how the topology of a neuronal network affects its activity patterns. And more specifically, we're interested in understanding how modular topology does that. And by modular, I mean groups of neurons which have a very high connectivity within the group and sparse connectivity between the different groups of neurons. So let me just start by describing our experimental setup. So as it was said, we work on cultures, and so we use multi-electrode arrays to record the activity. So these are thin film integrated microelectrodes that allow to record simultaneously the activity of many neurons for very long periods of time. And we use embryonic brains of rats. So we take the brains out, dissociate them, play them onto the MIA electrode, and then within a few hours, the neurons begins to send out extensions, connect to one another, and within a few days we have a network, and it begins to be active, and we can record the spontaneous activity of those networks with electrodes. So if you look at uniform networks, not pattern networks, but regular uniform cultures, when they are developing, they have a very stereotypic activity. So this is a raster plot where every row corresponds to the activity of a single neuron, and every black dot corresponds to a spike. And you can see that there is very typical behavior of network burst. That is this short event of intense synchronous firing throughout the network, and they are separated by longer events of sporadic firing. And there are two sets of neurons. You basically can see that some of the neurons also fire between the bursts, and I'll get into that a bit later. But generally, most of them fire within the burst, and you can just integrate over the activity of all the neurons and get the population activity. And you can see that it is typified by a fast onset and a more gradual decay. Now, those networks in cultures were extensively investigated, but all of this work, or almost all of this work, was done in uniform networks. By uniform, I mean networks where the cell bodies are uniformly distributed over the two-dimensional surface, and their extensions, the axons and dendrites, are not restricted in their directionality. So they spontaneously connect, maybe not randomly, but spontaneously. And the way that you make such cultures is just by applying an adhesive substance on the surface, which limits the motility of the cells and doesn't allow them to move too much, and you end up with a uniform conformation at the end. Now, if we look at a small section of the network, and this is imaged with a calcium indicator, so you can see that the connectivity is very intricate. And if your aim is to study the relation between the activity and the connectivity, it's very hard to do it in those kind of cultures, because you can easily see that saying who is connected to who is very problematic here. So because of this reason, a lot of effort was invested into trying to control the connectivity patterns of networks in culture. So the ultimate goal was to get networks such as this, where you have neurons at the nodes of a grid with extensions connecting them along grid lines. And a very nice practice was to couple such networks with multi-electrode arrays. And then you can potentially get a recording of almost all of the cells in the network, and the connectivity is mapped. And those work, indeed there were a lot of nice patterning techniques that were developed, and very nice images, as you can see here. However, if you look into those papers, you mostly find that they investigated basically only the capacity, showed only the capacity to stimulate and to record. And they did very, very little activity analysis. And I think that the reason for that is that there's a problem here of stability and yield. If you take and seed with very low density, then the cells either do not connect or are not spontaneously active. And if you increase the density, then the cells begin to exert pulling forces on one another, and eventually your pattern is ruined, and you end up with those clumps or clusters of cells which are distributed in different places in the culture. So we took a slightly different approach. We said, okay, the cells like to cluster, so let's use this innate propensity of the cells to cluster in order to try and build these modular networks where you have, again, groups of neurons with high connectivity, which are sparsely connected to other groups. And then we wanted to see what happens when we make this transition from a uniform network into this modular topology. So using different network patterning techniques, we were able to create 3 main network models and compare their activity to the uniform network. So I will begin by showing some results from isolated clusters, which are groups of highly connected cells with no connections sent or received from the outside. Then I'll show you what happens when you couple 2 of these together through a bundle of extensions. And finally, I'll show some results from networks of connected clusters. So I want to say a few words about patterning techniques. Basically, there are 2 ways to pattern neurons onto flat surfaces. The first way is by utilizing the surface chemistry. We specifically use soft lithography in order to pattern those islands of poly-D-lysine on the surface to which the neurons later adhere, and we can record their activity. The other way is to use rough surfaces. So it turns out that neurons love rough surfaces, probably because this is more similar to what they see in the brain. And we specifically used electrodes that we manufactured from carbon nanotubes that served the double role of both anchoring the cells onto the electrode and facilitating the electrical recording. So this is basically the 2 methods that we used. And yes?
Why not just take a large uniform network and then carve the activated field channels and isolate them?
Okay, so if it's a large uniform network, so it has many cells. If you want to specifically target the cells and the...
You activate all the cells except for a few items.
Yeah, but how do you deactivate specific areas within a network, right? All this is sitting in a dish with medium, and you want to approach a specific cell. So you either go cell by cell and do some kind of manipulation. Let's say you can, for example, use genetic manipulations and activate them with light or things like that, but you need some way to access specific cells. And even if you access specific cells, it's even much harder to control the connectivity or who is connected to, because this is formed randomly in the network and you have no control over it. So to give you some insight on how those networks self-organize, I want to show you a short movie. So here in day 0, the cells were uniformly plated, and as time goes by, they begin to send out extensions and connect to one another, and they exert pulling forces on one another. And there's a very interesting mechanics that is going on here. And in this case, we pattern big islands of poly D lysine on top of the electrodes. So eventually, due to the pulling forces, the islands anchor onto the poly D lysine clusters, and then we can record their activity. So these clusters are very big. They are clusters of about 500 cells. But we used clusters of different sizes, and I'll talk about it a bit later. Basically, you can't control really the number of number of neurons, but statistically you get different numbers of neurons, and you can count them, and then you can have recordings of different sizes. All this movie was 7 days. Yes. Sorry, I didn't hear your question. Why they are exactly around the electrodes? Yeah, so as I mentioned, what we did, we patterned ear islands of poly D lysine. Only ear, and ear, and ear. Okay? So I don't want to get too much into the organization of those clusters, but I just want to say a few things. So this is an image of one cluster. The synapses are stained in red, and the glia cells, the astrocytes, in green. And this is a cross-section, and this is a cut in the z-axis of a different cluster. And you can appreciate that the connectivities here are very intricate. So actually, now each cluster is different. It has a different number of cells, different ratio of excitator and inhibitor neurons, different connectivity patterns. So this is an important point. And also, if you look at connected clusters, you will see that those connections, those bundles, have both glia cells and dendritic axons. I want to start with showing results from isolated clusters. So this is a voltage trace recorded from this isolated cluster, and as you can see, each electrode in our setup records the superimposed activity of many neurons. So we can take these traces and translate them into the activity intensity of the cluster and present it in color code, because we have many such electrodes within each MIA dish. We can record them simultaneously, and if you do that, you get these activity traces. This is from 3 separate clusters. And the most prominent feature that you immediately see, that again you have these bursts, very similar to a large uniform network of hundreds of thousands of cells, but here we have only a few tens of cells. And you can also see this fast increase and a more gradual decay in activity, and you can measure statistical parameters, for example, the width of the burst or the interval between the bursts. And if you do so this is the distribution of interburst intervals for one specific cluster, and you can see that it is characterized by an asymmetric distribution with a typical value of about 10 seconds between the bursts. And if you look at the width, then you have a more symmetric distribution with a typical value of about several hundreds of milliseconds. And this is very, very typical to what you see in large uniform networks. However, as was mentioned before, the nice thing about our setup is that we can look at the activity of clusters of different sizes, and then we can see how the activity scales with the size of the network. So what you see here is the average burst rate, and by looking at such plots, there are several interesting features that you can observe. For example, clusters of about 40 cells already show this bursting activity. So 40 cells is enough to sustain these activity patterns. Now, if you increase the size of the network, you have a slight increase in the burst rate and in the burst rate width, but this increase is not significant. Nothing dramatic changes when you change the size of the network. And actually, clusters of about 100 neurons already show similar statistical features to large networks of uniform networks of hundreds of thousands of cells marked here in black. Another thing that you observe is that if you go to small clusters below 20 cells, you don't get bursting activity. You either don't get any activity or you get only single spikes. So this means that maybe there's a minimal network size that is required to sustain those profiles. Yes. Yes, so we didn't check the percentage, but there are excitatory inhibitory synapses. Before I showed an image with, of all of them. And in terms of excitatory inhibitory neurons, the ratio in those cultures is about 1 to 4. Okay, so there are very similar things between the uniform cultures and the isolated clusters, but we've also measured things that were previously unobserved in uniform networks. So what you see here is 500 consecutive bursts taken from one isolated cluster. So we just collect the bursts, we put them one below the other, and then we need to align them one relatively to another, and then we can see those activity patterns, and if we average, we have this profile, and this profile, as you can see, is characterized by again, this fast onset and the gradual decay, but on top of that, there is a clear oscillatory activity. Now, if you look at the frequencies, in this case, it corresponds to about 60 hertz. And if you look at many clusters, so the...
Sorry, what you did was just line up the beginning of the curve?
No, so what, because if you, for example, apply simulations, so you have a clear onset in time. But because this is spontaneous activity, you can't just say when the burst began. So what you do, you take traces, okay, and calculate the cross correlation between many pairs of them, and then you put this into an optimization algorithm that maximizes the total correlation in all the bursts.
So, do these bursts actually travel across the cluster in some interesting way?
Okay, so it's a good question. So the important thing about the bursts is that they are collective. Okay, so these are voltage traces, raw voltage traces from 5 consecutive bursts. And if you look at each oscillation phase, you can see that the exact spike timings are different in each burst. However, as a group, they maintain the same oscillatory behavior. The percentage of cells. So I can't tell you that. We try to do it by using calcium imaging. However, in those clusters, because the connectivity is very dense, it all looks like one big blob of light. It's very hard to separate them. I would assume that all of the cluster is active. If you look at small uniform networks, you will, and there you can image quite good individual cells. You see that all of them are active, or almost all of them. Now, another thing that I want to mention here is this frequency range of 20 to 100 hertz, which corresponds to the gamma band. And in this respect, we block inhibition with acutylene, and this abolished the oscillations, so they may be of the same source. So I just want to quickly sum up this section of the isolated clusters. So we've seen that you can take networks of a few tens of cells. Those networks exhibit network-wide bursts, which are typical of developing networks, and they also have oscillations. But the nice feature is that if the cluster is big enough to sustain this bursting activity, this activity doesn't change much as a function of the cluster size. And I think that this is a bit surprising if you take into account the fact that every cluster in our setup is different. So again, it has different ratios of cells, different connectivity patterns, but yet they all sustain the same activity, and this suggests that there are self-regulating mechanisms that do those things. And I want to show an example from a different study that utilizes patterned networks. they did was they patterned 4 sizes of uniform networks, and they examined different morphological properties of these networks. So they noticed that if you look at synaptic density and the dendritic density in those cultures, you can see that the large cultures have higher synaptic density and dendritic density, which means they should have higher levels of excitability. However, what they also noticed is that if you examine the synapses of excitatory neurons onto inhibitory neurons, then you have much less synapses in the small network. And if you look at the excitatory postsynaptic currents, then they are larger in smaller networks. So these two contributions compensate over the increased excitability due to the size. Now, another example is from a study that we did several years ago. And if you remember, I noted those cells that fire between the bursts that are more active than the rest of the cells in the network. So if you look at the fraction of these cells in situations where the activity levels are very low in the network, for example, at early development, you will see that you have much more cells like that, and their fraction is reduced as the network matures and the activity intensifies. And you can do the same if you block the excitatory activity, and then again you get a rise in the fraction of the cells. And when you wash it, you have a gradual decay almost back to baseline levels. So again, there are different mechanisms that the network employs in order to sustain those patterns. So now, what happens when we take two such clusters and we couple them? So this is the activity profile, the activity intensity of those two clusters. And what you can see is that similarly to uniform clusters and to clusters and uniform networks, you have these bursts that span both clusters. However, in contrast, you also have these bursts that do not propagate to the next cluster. So you have a burst that is initiated here and does not propagate. And this means that there is some conditional activation here. Now, if you look at the burst that did propagate, so this is an example of one burst, you can see that cluster 1, the left one, was activated first, and it activated the second cluster. And if you look at those delays, you see that they are several tens of milliseconds, which is a lot. It's on the time scale of the recruitment time of the whole network, both in the isolated clusters and in the uniform networks. So there are long delays. And in this case, cluster 1 fired first, but if you look at many bursts from both clusters, you can see that this is almost always the case. So there's, in most cases, a dominant cluster and a slave cluster, and the dominant cluster activates the slave. Yes?
So how strongly are the clusters coupled?
So what is the width of this connection? Okay, so I'll get to that. I can't say exactly, but I'll get to how it influences the activity. And if you measure again the oscillations, so we have those oscillations in both clusters as in the isolated system. So I want to just go now into those properties in detail. And I want to start by looking at the asymmetry. So this is one of the first systems that we measured. So here you have 3 clusters connected in a chain. And if you look at 100 consecutive bursts in each of the clusters, you can see these activity profiles where mostly cluster 1 is the first to be active, and then its activity propagates to cluster 2 and cluster 3. But if you do a more detailed analysis, you see that it's not only the activity does not propagate only to one direction. So what you can take is you can take every burst and construct the spatiotemporal profile of this burst, then take consecutive bursts and correlate this profile with consecutive bursts, and then you can build the matrix where in every cell in the matrix you put this value of correlation. So this is what I did here, and I just ordered the matrix with the dendrogram. You can clearly see that you have 2 groups of spatiotemporal patterns. pattern when activity starts from cluster 3 and propagate to cluster 1, and this group to the opposite pattern. Now, I want to mention that here I also only took the bursts that activated all 3 clusters. Of course, there are bursts that do not propagate, as I mentioned before. But you can see that there is a very large asymmetry, and this is the case in most clusters.
Don't belong to any of these.
Most of the bursts don't belong to any of it. Most of the bursts are isolated, actually, and I'll talk about it later also.
So do you stimulate the separately, or is it spontaneous activity?
Yeah, so we can stimulate, but this is all from spontaneous activity. Okay, so another thing that we can check is how the topology of the network influences the functional symmetry between pairs of clusters. So what we did here is we took pairs of clusters within a chain that are embedded in a larger network of clusters. And we had coupled clusters, and we had chains from which are marked here with those dots, and we had sometimes at the end of the chain these residual groups of clusters which are marked by a letter R. So here I show an example of this network where we have 2 coupled clusters which are coupled on the right by this large residual group of clusters. And you can calculate the cross-correlation function, and this is shown here, and you immediately see that it has a peak at -40, which means that activity mostly propagates to this direction. Now, in order to quantify the asymmetry, you can just take the integral over the left side and subtract it from the integral over the right side and divide by the sum of both, and then you get a measure between -1 and 1 of the functional symmetry here. And the interesting examples are to look at functional symmetry in cases where structural symmetry is where the structure is asymmetric. So if we look at this network where we have 2 coupled clusters and a chain of clusters on one side, then we will measure usually negative symmetry, functional symmetry, which means that activity propagates upwards. If we further increase the asymmetry by adding this residual group, then the functional symmetry is even higher. And if we go back to a more symmetric case, where we have a chain on the other side, the symmetry is reduced again. So the structural symmetry of the embedding network affects the functional symmetry of the pair of connected subpopulations. Now, I want to show you some results that relate to the conditional activation that I described before. So here again, we took those chains of clusters, and each time we took a pair of clusters, and we try to look at the intensity of activity in the receiving cluster as a function of the activity in the sending cluster. So, as I told you before, activity goes to both ways, although one way is more dominant. So we have to define the sending cluster. So we just do it by taking only the bursts that propagated to the right in this case. So we take all these bursts, and then we reorder them according to intensity. So we have the low intensity burst at the beginning and the high intensity burst at the end. And if you look for this burst, you look at the response at the receiving cluster, you can see that for weak burst you don't get any response, and for a stronger burst you begin to get responses. And you can see the same picture if you look at the probability of getting a certain intensity in the receiving cluster as a function of the sending cluster. And here again, for a weak burst in the sending cluster, we don't get any response, and as we cross a certain threshold, we begin to get responses in the receiving cluster. Now, I should mention that it's not always a threshold that you have to cross, but the general rule is that stronger activation of the sending cluster will yield stronger activation of the receiving cluster. And this is quantified here by just taking many pairs and calculating the correlation over such plots, and you can see it's almost always positive.
So the yellow spots here is noise, or it's
no, this is a probability plot. So the light light spots say that there is a small probability of getting a burst with this intensity in the receiving cluster and this intensity in the sending cluster. And altogether, you see that all those, most of the bursts are with the high intensity in the receiving cluster are concentrated here, which means that they also have a high intensity in the sending cluster. So we've seen that there is a conditional activation between connected subpopulations. Yeah.
So question? In previous experiment, were there exactly two clusters, or were you only studying those two clusters?
No, so each time we take two clusters in a chain. So we have those chains, and there can be a network here and a network here, but we always look at those two.
Are those other clusters also connected to these two?
From the side. But also the same, you can see the same phenomena when you look at just two clusters with no clusters on the sides.
So, I mean, could it be that the third cluster is simulating both, except that it has different delays and that's what is causing it?
No, so this is a chain. The thing is that you always look at chains. So only one-dimensional. There can be something here, but okay. So a question that you can ask, we have this conditional activation, but you can ask yourself whether you can control it in some way. And then you have a gate. So we specifically try to see if we can use, can break the balance between excitation and inhibition in the network in order to control this conditional activation. So here I show results from this network. It's a bit hard to see, but we have a chain of 5 clusters, this chain, and we can measure its activity. So these are the activities in the control conditions. We have the traces, the activity of different clusters, and you can see these bursts in individual clusters. However, you can see that those bursts do not always propagate, or most of the time they do not propagate to connected clusters. Sometimes we have these propagations, and sometimes we have larger bursts, but most of the time they do not propagate. Now, if you take bicuculin and you block the inhibition in the network, you have a transition to this state where suddenly you have this burst that span all the network. So the segregation that existed here disappears completely, and you have only this integrated activity. Now, you can also look at the propagation pattern if you zoom into one burst. So this is this burst in this case, and here the burst started at cluster 3 and propagated to the left and the right, and you can present this propagation by this schematic drawing where a blue dot corresponds to the firing of the cluster and the arrow corresponds to the directionality of propagation. And again, you can see that in the inhibited conditions with the bicuculin, you see a very ordered propagation between the clusters, and all of them are activated. You can also see the delays get much shorter in this system. Now, you can plot this schematic drawing for many consecutive clusters, and then again you see the same image. Most of the clusters are activated alone, and the synchrony does not propagate, and sometimes it does, but once you remove the inhibition from the network, you get those profiles where all of them, or almost all of them, start from cluster 5 and propagate to the rest of the network. And I think this is also interesting if you think about in the context of epilepsy, because suddenly you have this focus that emerges in this network.
This looks very much like what Aviv calls a SynFire chain.
Okay, so in terms of SynFire chains, this would be a very nice setup to look at SynFire chains, I agree. Specifically, what we examine, I think it's a bit different, because if we take one cluster out of the system, so I think a SynFire chain will be the sequential activations of different groups of neurons within this cluster, and the activation of the different groups in the chain has a relatively short delay. So here what happens when activity propagates, it's actually a recruitment within the cluster, and when this recruitment is built and crosses a certain threshold, then you can see the activation of the next cluster. So I think it's a bit different than thin-fire chains, but yeah, I agree that it's a very interesting direction to try to look at it this way. So I want to show you some results now from a clustered network. So obviously there are interesting things that happen when you switch from uniform topology of the network to a modular topology. But the question is, what happens if you enlarge the system and go to a network with many connected clusters? So I want to start by showing you a movie of the activity. Here it's a bit hard to see the clusters, but this is the physical space of the electrodes. Activity will be shown as elevation in the z-axis. So this is on a long time scale, and you can see that the bursts are initiated in different places in the network each time, and they propagate to different sizes of the network in each burst. And if we look at one specific burst, then you can see that the activity is typified by sequential activation, where clusters first initiate their nearest neighbors and then propagate to the rest of the network. Another feature here is the recurrent activation loops, where within one burst, a single cluster can be activated more than once.
Is each cluster only connected to the nearest neighbor?
Sorry?
Is each cluster
Yeah. Obviously, this is due to the connectivity patterns.
They are only connected to the nearest neighbor?
Okay. So they are mostly connected to the nearest neighbors, but they are also connected to the next station. The way that you can find this out is if you silence the network and you stimulate it. Electrically stimulate the clusters. And then, after a short latency, after the stimulation, about 5 milliseconds, you can get a response in single neurons, and later there's the bursting response of the whole network, and you can see it also in the next cluster. More than the next, I never saw something like that, but almost all the connections are nearby.
So again, this is not spontaneous activity, right? You don't stimulate clusters.
Yeah, all spontaneous. So looking at the same network that I have shown you before, these are the activity patterns in this network, and you can see, again, the same features where you have bursts of different sizes, smaller and larger, and they start from different places in the network. This is very different from what you've seen in the uniform cultures, where, again, you only see this activation of the whole network together, and you don't see anything else. And if you zoom into one of these bursts and reorder the electrodes according to the center of mass of the activity, then you see again the sequential propagation, and you also see the long time delays, because, for example, the last cluster here is activated long after the first cluster has finished firing, so the delays accumulate, and you get eventually very long delays. And you can also see the sequential activation if you plot this burst on the physical space and mark the center of mass with color code. And here, in this case, it started here, propagated to the left, and then upwards. And this is for one specific burst. Different bursts have different propagation patterns. And you can also look at a correlation between each cluster and all the other clusters. So if you take for each one only the most correlated cluster and plot it on physical space, you will always get that the strongest correlation are between nearest neighbors. And again, this is for the same reason, because of the sequential propagation. And if you measure it for all cluster pairs, you'll get this decrease with distance. So another question that you can ask is, what happens in terms of unbalancing the inhibition in this scenario where we have a large network of connected clusters? So obviously, when you have a two-dimensional network, you cannot just calculate the cross-correlation function as we did with the chains. So what you can do is take one cluster and all the adjacent cluster to this cluster. And then you can calculate the cross correlation between those two, and you can plot a vector which direction is in the direction between the cluster and which magnitude corresponds to the cross correlation function, to the time. And then you can get an average vector. Okay, so this is the blue vector here, and you can plot this vector for all the clusters over many consecutive bursts. And this is what is shown here. So if you look at these plots, you can see that the distribution of angles is very wide, which means that activity flows here to different direction in every burst. However, when we switch to the bicuckoling conditions, where we inhibit the inhibition, you can see the distribution is much narrower. And you can also calculate the average vector from these blue vectors, and it is marked here in red. And then you can see again the focus, that in this case starts here and propagates upwards and to the right. So also in these two-dimensional clustered networks, we have such focus after we remove the inhibition. Okay, so coming back to the question about the width of the connections between the clusters. So indeed, there are interesting features that emerge when you change the topology and you introduce this modular structure. However, you can ask, how wide should the connection be? Or what is the ratio between the connectivity within the cluster and the connectivity between the cluster that gives rise to those features? So what we did, we looked at this uniform network, and here we have again these bursts that span all the network, and we also looked at these clustered networks where we have clusters with very dense connectivity between them. And what you can see that we have basically the same pattern. So also here we have those bursts that span all the network. So it's not enough to have clusters. It's really the ratio between the connectivity within the cluster and the connectivity between the clusters that matters. And only by moving to this setup where we have loose connectivity between the clusters can we get those the features that I described. Now another point that I want to stress is that, again, it's not the clusters that are important. We chose the clusters as a model because it's convenient to make such networks. However, any break of the network uniformity will give you the features, and in a different study that we did, we took two large uniform networks, and we coupled them through a thin bridge, and then we also observed part of the patterns that I described. So this is all in terms of the results. So let me just summarize everything. So we've seen that you can utilize the self-organizations of neurons and glia in culture to make those networks of clusters, the isolated clusters and connected clusters. And if you look at isolated clusters, you can see that their activity is very similar to large uniform network. And they are characterized by this fast onset of activity and the short delay between the neurons. And we've also seen that there's a minimal network size that is required to sustain those bursting activities. However, once you cross this minimal network size and you begin to increase the size of the network, then you cannot change drastically the activity patterns. They maybe change a bit, but they do not change much. And this means that by adding cells to this network, you cannot do anything dramatic. Only by breaking the uniformity of the network, and we did it by implementing those clusters which are loosely connected to one another, and you start gaining new and exciting features. And those features include long delays. And if you think of long delays in terms of information processing, then they can be very helpful, because if you want to separate in time across different processes, then those long delays can be very important. Also, we've seen that there's an inherent asymmetry between pairs of clusters, and also that this asymmetry is affected by the topology of the embedding network. So it can be important if you want to control the activity flow through the network. Now, in terms of the conditional activation, this gives you a very important tool, because in those networks you have a very wide spectrum of activations, from segregated activation within the subpopulation to integrated activation of all the subpopulation, and not only the integrated activation that you get in the uniform network. And also you can use the unbalancing of inhibition, for example, with some neuromodulator, and to achieve gating between connected subpopulations. And finally, what I want to suggest that this model in culture can be a very useful system to study information processing in culture, because each module or each cluster in this system has all the richness of activity patterns that were previously described in uniform networks. However, by coupling those clusters with loose connectivity, we get new and exciting features. So this is one of the points. And finally, I want to thank my supervisors, Professor Eshel Ben Jacob and Professor Yael Hanin, and the lab members and the technical staff at our lab, Ina, and thank you for listening. Sorry. 4 excitatory, 1 inhibitory.
You mentioned that when you have an isolated cluster, it has a bicyclin, then you eliminate the bursting.
No, you do not eliminate the bursting, you eliminate the oscillations. Okay, so let me just show.
Is that the secondary oscillation?
Wait a minute, I just have a slide so I can show you directly. Okay, so this is the burst with the oscillations, and this is the profile. And once you block inhibition, you still get a burst, but then it will have a sharper onset, and the decay will be longer. Okay, so the burst will still be there, but you won't get any oscillations in the cluster.
So what's the intuition for this burst? Where did it come from?
So there are many there are many theories about that. I think that the most based theory is that they come from integration of synaptic noise, because those cultures don't have any input. So
Basically a critical phenomenon. So it's an avalanche.
yeah, so there's the work of Beggs and Plant to exactly look at such cultures as avalanches, and yeah.
Do you have any intuition what would happen if you had longer connections between clusters, so not just
longer in terms of the length of the extension? So I don't think that the length of the extension plays.
They don't form connection only to the nearest one, but also farther away.
So they move, but mostly they do not move too much ahead. But the more connections you add to the network, the more it looks like a uniform network. Okay, because everyone is getting connected to everyone, so you will get more closer to the uniform profiles. And in terms of the length of the bundle, I don't think it matters too much, because those are fast propagation relative to.
So I'm trying to see the long-term goal of the studies that look very elegant in the way you can generate machines that work by neurons, but
There are two possibilities. One is that we have access to the brain, and the other that we achieve some technologies. And I just saw that you have a proof of concept that you can have networks, and they can have some phenomena which are related or not related to the brain. Where do you think the lab or you go to in these two directions?
Okay.
For example, trying to stimulate, trying to teach, trying to change, etc. What are the possibilities?
Mm-hmm. Okay. So there are some people that do plasticity, for example, with those with such cultures, and they try to teach them. So I think that our setup specifically is better than other setups in this sense, because let's say that you have a uniform culture, and there is the work of Shimon Maron from the Technion, for example, where it tries to use dopamine and electrical stimulation in order to induce some kind of plasticity. So if you just take a random network and you stimulate it at different places, then you cannot really control which neurons get potentiated, because you stimulate a lot of neurons and you don't have any specific connectivities within them. However, in this system, if, for example, I take one subset, one subpopulation, excite it or stimulate it, and then wait 5 milliseconds and stimulate the other subpopulation, then you get a clear time delay between the population, and then you can look at the plasticity at the population level. So this would be one benefit of this system. I think that the most important benefit in terms of studying, of relating to the brain, and we don't use in the lab the word brain much, because again, these are still artificial systems. I think that in the brain, when you want to study the topology or the effect of topology on activity, it's very hard, because to extract the topology out of this mesh of circuits that you have in the brain is something that, I don't know, I think it's in most cases impossible. And here you have an opportunity to really isolate this effect of topology on the circuit activity. Now, you can say that the conditions in this case are more relevant, for example, for developing a network, and not for a cortical network that show asynchronous irregular activity or something like that. But first of all, there are ways to make those networks show activity that is not bursting, for example, by application of carbachol. So this would be one way. And I think another thing is that people are doing is there's the work of Steve Potter, and they are trying to use simulations in order to make those irregular firing patterns. And the motivation is that these cultures do not receive any input from the outside. So they are, let's say, in a mode of sleep. So if you constantly, from day zero, give stimulations to those neurons, they eventually develop slightly different activity patterns. However, there are arguments about if you can do it or not. There's one work by Daniel Wegener that shows that he did it, and I heard other people say that it's problematic, but I guess it's something that you can try. Yes. Okay, so we looked at the resolution of 100 neurons. What I tried to say is that if you go beyond 100, I try to say that there is no point of going beyond 100, because you practically get similar statistics. Of course, this is not true, because when you have more neurons, you have more complexity. But in terms of the overall activity of the whole network, the population activity, the statistics are not very different, and you still get those bursts, and even in the same range of rates. And
You know what was the average connectivity within this platform? How many nodes are actually connected to each other?
so they are very, very densely connected. If you remember the image. Sorry?
It's really like infinite dimension.
Yeah, I guess.
So beyond that, the statistics is more or less stabilized. This is something like a granular. I would actually expect some sort of multiscale because it's very large, not just 100 and large, much more than that.
So in terms of multiscale, there's a work actually from Eshel's lab where they checked the interval between the bursts, and they showed that they have a Levy distribution, so they have a heavy tail, and when we looked at single clusters, so I may have an image of that here also. Okay, so
What was this term?
yeah, so this is what I want to show is some. So this is on the time scale of milliseconds. So this is 5 seconds altogether, and this is one burst, and this window is the small window here on the longer time scale, and all this window is the small window here in longer time scale, and again in a longer time scale. So as you can see that this activity changes on very different time scales, and this is one of the benefits of this system, because you can really record for many days. And so we see also.
Same as Shein Alright. Look at isolated models that the width of the IBI distribution goes down with model size.
The width of the ISI distribution, the IBI distribution. Yeah, it should. I have to say that I didn't check it, but this is my guess. I think that you know what? It may go. It also may go up, because if you think about it, as the smaller the system, the less it is stable. So in essence, it may, because if it's larger, the statistic is cancelled. You know, you have the central limit theorem. So this is my guess actually, but I have to admit I didn't check it.